Hi everyone. So it's Tuesday, the 21st of April, and I promised that I would give you a tutorial today on primer design for DNA metabarcoding. Um, and I'm going to start out by saying that there is no one way to do this. This is probably the most frustrating thing for you to hear about the whole process, but there are hundreds of different ways to do it. I'm going to go through the way I do it mostly, which is actually mostly manual, and it's done by moving between various different websites and just one or two bits of software that are free and you can download if you want to give them a go. And hopefully that will be a reasonable way of describing the overall process so that you get an idea of it. There's a bit of an art to it, and there's lots of testing involved. But anyway, I'm going to start out with a hypothetical situation. So this is a picture of a pangolin on the screen here, and you will have heard about these mammals because they were potentially linked to the current coronavirus outbreak because a similar virus was isolated from pangolins. It turns out that they probably also caught it from bats, as we did, most likely. That seems like the most likely explanation. So pangolins are mammals. They're the only scaled mammal, and they are the most traded mammal in the world because of their use in East Asian traditional medicines. They use their scales ground up to, well, not to do anything at all, actually, because it's mostly just keratin, but supposedly to do useful things. So it's really rough for them. They're very highly endangered. They occur all over. This is just a Wikipedia page for it. They occur, thought there was a map, but here we are. They occur throughout Asia, although of course their populations are heavily hunted there, and also there are several species in Africa, and a lot of the wildlife trade is between these two parts of the world. So that creates a situation where we might want to design a PCR primer set for identifying their parts. So if you found a pangolin scale at an airport, if you were part of the biosecurity team of any country that had captured one of these, either the products of these species or the actual animals themselves. You might want to identify which species it was because you can see they have actually restricted geographic ranges to some extent anyway. That might help you get an idea of the provenance of the material that you're interested in. So you would maybe want to design a PCR primer set that focused only on pangolins so that you didn't get any confusion with other types of species and excluded the DNA of other organisms. So let's go ahead with that hypothetical example, and I'll show you how I'd go about it. So I know that pangolins are a mammal group, so they're here on this cladogram phylogenetic tree, um, moderately closely related to carnivora. I know from looking at other things that this division is only about 60 to 70 million years old. So that means it's the sort of level of variation that we might be able to find with mitochondrial markers rather than nuclear markers. Um, so this is part of the problem with this whole issue. It's knowing which gene region to look at in the first place, and to a large extent that is determined by the rate of evolution of things. So actually, the very first thing I'd do in a situation like this, if I was going to start designing a new group-specific primer set for a group of interest, would be to do a web search and find out if something else is available. So I already did that earlier with a search for, where's the page, um, for tardigrades. So I think I've mentioned tardigrades in my talks before. These are water bears, these tiny microscopic animals that can survive in space and so on. And I would just do a web search for group-specific primers and tardigrades. So I'll just show you what I did. So I Google search for group-specific PCR primers for tardigrades, and look, there, someone's already designed some more than 10 years ago. So that's actually the first thing you should do is go and look for other primer sets, have a look at the paper that describes them because people tend to publish these things and see if they're useful for your application. So In this case, because tardigrades are a very old separate group, they're actually their own phylum. They looked at the 18S nuclear DNA region. These are just some gel images showing that they did some empirical testing where their PCR primers amplified from tardigrades, but not from other groups. And some phylogenetic trees showing how things are related to each other. But of course, they had a reasonable idea that they needed to use a slow evolving molecule for that. And of course, it's difficult if you haven't been working in genetics for a long time to know which molecules evolve at which rates. There's no real database for anything like that. I mean, I have talked about the International Barcoding of Life database and the cytochrome oxidase 1 mitochondrial DNA sequence. And that's been used extensively and can even separate between most species of living animals. So if you want to look at closely related species of insects or crustaceans in the sea or something like that and tell them apart, then this you would think would be a reasonable way to do it. But unfortunately, the whole project was set up before DNA metabarcoding. It was set up for DNA barcoding. And the region that they use, the cytochrome oxidase 1 region, is variable all the way through it because it's a protein coding gene. I'll explain the reasons for that in a moment, why that's important. Well, I have explained it before in lectures, so I won't go into it too much. But basically what it means is that if you're trying to design a group-specific primer set, like we are going to design for pangolins in a moment, there's no way that you can find invariant PCR primer binding sites. So there'll always be some variation in the binding site, and that makes it very difficult. So I tend not to use this region. Other people do for DNA metabarcoding. It's a personal preference to some extent, but of course I'm right and they're wrong. So other ones that you can use, of course, are the ribosomal RNA genes. There are some in mitochondria, which do evolve at a similar rate to the barcoding of life CO1 gene. So you can use the 16S region or the 12S region in mitochondria, and you can have primer binding sites that are conserved and will allow you to have some certainty that you're going to amplify the things you want to amplify. For more slowly evolving things like the tardigrade version, there's actually a whole database dedicated to this. So this Silver database is a database for the two nuclear ribosomal RNA genes, the 18S and the 28S. It's actually very good. It's got a lot of information in it, and it does actually have a primer search function in it. So if you are looking for designing a primer set that will amplify from, say, one phylum and not from others, then this is the place you go. It has potential ways that you can design primers here, or at least test them on everything in the database. But and it has some examples down here, which you can go through if you want. Anyway, I recommend having a look at that database. It's certainly, if you want to get more knowledge about this area, this is a very useful resource. Anyway, back to pangolins. So if I was trying to design a mitochondrial DNA primer set for pangolins, what I'd do is I'd go to the genome database, just type pangolin in here, because I can't remember any of their Latin names. I don't know. I only just thought of doing pangolins 10 minutes ago. If you type that in, it comes up with all of the genomes that are available for pangolins. And of course, the mitochondrial genome is the one that's most often sequenced in animals. Lots of mitochondrial genomes are available. It turns out, and the mitochondria is an organelle, so that's what that means there. So it turns out there is a mitochondrial genome available for pangolins. So let's have a look at that. This is the NCBI GenBank database. That's the organism, that's the project, these are replicons. So if we just click, basically just click on things until you get what you want, we get to a mitochondrion. So here we are. Here's the complete genome of the mitochondrion of a pangolin, one off. So what you can do is you can scroll through that. Mitochondria aren't very big. They're only about 16 kilobases long in mammals, 16 and a half there. So that's the whole thing there listed out. But as I was saying, Ribosomal RNA genes are pretty good for designing PCR primers, just because they have conserved regions, and there's the cytochrome oxidase 1 gene. If you want to use that, you can isolate that. But let's try and find the mitochondrial 16S gene. That's what I want. So the 16S ribosomal RNA is listed here in this list. We can go to it there, and I'm just going to change the display type to FASTA, which is the sort of no annotation, very simple thing. So this is the 16S ribosomal RNA gene isolated here. So this is pretty neat. This is something you can actually start working with manually. So I just do this manually, just because there are software options for this, but I just find it easier to do it this way. Maybe it's because I'm kind of old-fashioned. I've been doing this for 20 years, but it sort of helps you hopefully to see how it works. So I'm going to just save this notepad file. So notepad's just a text editor. You can use different things in Macs if you use those, or Linux, which you will be using next week once I finish this class, which I will be talking about extensively next week. So I'm just going to save this on my desktop, and so it's going to be pangolins. Right, so that's on the desktop now. But because we've got that there, GenBank's got all these great options. It's already got that sequence kind of highlighted. So what we can do is we can blast this sequence. So we can blast is the search tool that allows you to compare any DNA sequence to all the other DNA sequences in databases, in this case, all the DNA sequences in the world, essentially. So we can compare that to everything else that's in there. It's just using the name of it there rather than printing out the whole sequence. You could cut and paste it into there, but it's just done that subrange and the name. The database we're interested in is just the standard database, so that's everything. I think we're just going to leave all that as it is, and we're going to look for somewhat similar sequences. So let's blast that. And this may take a minute or two to come back, so I'm just going to pause. Okay, so that took a couple of minutes for that search to complete, and then it just automatically comes up with this page here. And on this page, there are a bunch of descriptions of hits. So there's a lot of information on it. So this was the ID for the thing we were searching with. This is the job title, so it keeps sorry, that's the ID of the query, what it was. It's nucleic acid. We blasted sort of 10% of the mitochondria in there. And it comes back with all these hits. And we can see that because these have the same genus name as the thing we were looking for, so this is these are closely related pangolins, or they're pangolins anyway, they're all in the same genus. And we can actually check the taxonomy. If we go to this tab, it'll tell us what they are. So yeah, so it's got 1, 2, 3, 4, 5, 6, 7, 9 pangolin species, and then close relatives of pangolins along here. And we know that carnivores and bats, in fact, are close relatives of these. So that's actually the perfect situation for designing a primer set for these. We've got all these things selected. I mean, it automatically selects those. So I'm actually going to download all of those. And what you can do is you can download the bit that aligned, so the fragments that we have in complete comparison to each other. So in this table of hits, it tells you query coverage, which is how much of the stuff aligned. And actually, most of those are really good. So most of these are coming from complete genome. So we should actually get a pretty neat overall alignment from this. Anyway, I'm going to align it. So I go down to align, the FASTA align sequences, and download that. The reason I do this, and we can actually look at them here, it's just that it gives you all the alignments individually. I want them all lined up together, and hopefully you'll see why in a moment. Okay, so that's it there. So I've got that file there, and I'm going to open that with a dedicated alignment program. The one I use is called AliView. So I'll just get the website so you can see what that is on this very carefully prepared tutorial. This is actually a great bit of software, completely free. You can download this if you want. And it installs on any platform. It's a Java application. So AliView, made by a scientist at Uppsala University. That's why it all sounds a bit Swedish. Anyway, good bit of software, very useful for this kind of thing. So I'm going to open that file that I just downloaded. And that is sorry, just getting to it. This is making fascinating viewing for you all, I'm sure. Right, I'm back. Sorry, that took me a minute to find that file. So once you open it up, these are all those sequences in this one big file. So I just go to the align menu here and I just say realign everything, and then it starts its process. Now I've got 100 sequences there, so it might take a little while once again. So I'm just going to pause. Okay, so I went away and made a cup of tea and came back, and this was finished. So that took 5 minutes or less to do. So what that did was it lined up all these sequences into what we call a multiple sequence alignment. So the names of the sequences are on the left here, all the sequences are on the right here. Hopefully you've come across something similar to this in genetics courses and other courses that you might have done in earlier on in your undergraduate phases. And these are all the sequences along here. And as you can see, there are regions of great similarity among all of them and other regions of variability. So this is the sort of stuff we work with to make all this happen. I could save all of this to a text file now if I wanted to, save it in Notepad or something like that so that we can repeat this. But the important thing to note is that it's sort of organized itself automatically phylogenetically. So these ones that I'll highlight up and down here, these are all of the pangolins. That's a horse. Those are rhinoceroses, pigs, and bats down here. So I sneakily looked those up while I was having my tea before I restarted the video. Anyway, all of these are the pangolins and the pangolin mitochondria, and actually highlighting that makes it a little bit harder to see. So I'm going to highlight the non-pangolins actually here. And we can go through the pangolins and find places where we might put good PCR primers. So what we want is 2 bits of DNA that are consistent within pangolins, but not present in or not perfectly present in non-pangolins, and that have a variable bit of DNA between them that we could use to identify pangolin species. So maybe a good thing to do is to look for a variable bit. So this bit here, this region, has got a reasonable amount of information in it. You can see that that's sort of messed up. It's got, so these two are from the same species here. So Manus pentadactyla, the top 2 are the same as you'd expect. But then there are 3 versions of crassicaudata here with some variation in them, but certainly consistently different from the pentadactyla. And yeah, it looks like, and Javanica is down there, and that's different from the others. So it looks like there's enough information there to delineate species. But even if we just amplify this short bit of DNA, and we might need to amplify from a little bit more just to make it good, we would be able to get a consistent answer there. So what we need to do now is find a consistent difference between these pangolin regions and all of the other things that are in here so that they don't get amplified. And another complicating factor to this is that once we start looking for those, we also need to start considering PCR primer efficiency. So we need to find regions where a good PCR primer would bind, and this starts to become complicated. It actually means you have a whole bunch of competing factors to consider when you're designing these primers, but I'm pretty sure we'll find something if we keep looking. We may need to look at other regions. That looks like we're getting a little bit too far away from those. I mean, there is, just looking along here, there is this T that's consistent in all of the pangolins, but not present in the non-pangolins, or the ones that we can see on the screen at the moment anyway. But because that's a T with a whole bunch of A's on it, I hope you remember from earlier lectures that A and T base pairs are weak compared to C and G base pairs. And we want the 3' end of our primers to fall on a C or a G if possible, or at least be CG rich in that area. So actually I don't see a good primer possibility there. So I'm going to scroll along a little bit further and see if we can find something that might be a bit better, a bit more pangolin friendly and unfriendly to other mammals. Where are we going with this? So once again, this would be a good species identification region. Good. It's starting to look all right. Yeah, it's a little bit tricky. Actually, let me continue. I need to jump into it right now. Let me pause the video for a minute. You can see that I'm thinking about this, and I'm going to scroll through this and come back to it in a moment when hopefully I've found some good regions. Okay, as you can tell, I'm completely winging this. I actually didn't plan to look for pangolins or anything until 10 minutes before I did this talk. But anyway, there is a region that's useful for us. So if you go through this alignment, you can see that at position, and I tend to write all this down on the notepad so that you can keep track of it. There's this C here that occurs in all of the pangolins and occasionally in some of the other mammals, but there's a better reverse primer. So at this C here, that only occurs in the pangolins. And in combination, that basically isn't going to amplify from anything other than pangolins if you use those two primer sites. So one primer will end on this C and amplify in this direction from left to right. The other primer, the reverse primer, will end on this C and amplify in the opposite direction. I've written down the primers that I would make to use in those two positions. So for the forward primer, it's just the same as the sequence we find here. So you can just basically cut and paste it. The only thing I had to do was, because there's some variation in here, so here it's sometimes a C and sometimes a T, depending on which species it is. So if you write that down as a Y, that's a code that lets the oligosynthesis machine synthesize either a C or a T in that position. So half of the oligos on average will be a C and half will be a T. The same thing for this position here, which is either a G or an A. You put an R there and it knows to do the same thing. So there's a standard set of codes for that. And this is the primer sequence we come up with here. For the reverse primer, it's a little bit different. So the reverse primer, the 3' end is there because it's going to amplify in that direction and along here. What you have to do then is make the opposite version of that, and that's called reverse complementing a sequence. So what I do is I copy that and I go to a website that will just do that for me. There are plenty of places, plenty of programs that will do this for you. So this is the sequence I put in, submit that, and it gives me back the reverse complement of it. And then we don't need that website anymore, so I'm going to close that. And go back to my notepad file, just where I'm storing all of this, and write this down. So reverse complement is that. So this, the C that was at the 3' end is actually on the other strand, it's a G. So now this is the 5' end of that. And the what you're synthesizing, although it's binding to a C, it's the opposite because it's priming off the other strand, so it's a G. So it's reading it in reverse. So from 3' to 5', it would be G, T, T, because the T's bind to the A's, C to bind to this G, A to bind to this T, and so on up until here, which is where I designed the primer to bind to. So that whole region. So those pairs together, once you've got those sequences, you can search that initial sequence that I cut and pasted out and find where it is. And I did that while I was shut off the video, so you didn't have to persist with listening to me do it. But that shows you how long the fragment is. You can cut that out of there, and it's 96 base pairs long. So it's actually a really nice small amplicon. It's got plenty of sequence variation in it that will amplify, that will identify pangolins if we can get pangolin material to amplify from it. So the next thing that you would do is check to see whether these PCR primers are any good. And essentially, that basically means whether they have reasonable annealing temperatures. And I use a website to do that. Once again, I use this website called OligoCalc. You can put oligos in here. Actually, I'll do the reverse primer. Oligos in here, and it will tell you what their melting temperature is. Oh, this also reverse complements things if you want to, so you could use this website too. Anyway, if you press this calculate button, it gives you all these options for the melting temperature of primers. It's saying that this is, and these are just different algorithms for doing it, saying that the reverse one has a 52-degree annealing temperature, which is perfectly acceptable. The forward primer, it's got a few redundant bases in it, and so that makes it a bit hard to tell because it's actually over a range, but it's 51 to 50. Well, some of the other estimates are even higher in that algorithm. But anyway, they're sort of roughly compatible. They're in the right sort of range. You can do this other thing, which is check to see whether they bind to each other. In that case, so primers can fold back and bind on themselves, or different copies of them can bind to each other. And it's not too bad for that one, and I should have done it for the previous one, which is this one, so I'll do that. Just check for self-complementarity. And basically, if they're really strongly self-complementary, then it's bad. This is not too bad. So there's a small region where this primer can bind to itself, but that's only 6 base pairs long, 7 base pairs long. So that's actually really not that bad. So we could go ahead with these primers. The last thing I would do, however, is just check, because when we did this alignment, of course, this isn't all of the DNA of everything in the world. These were just all of the most closely related mammals to the pangolins that we were looking at. I would just sequence I'll just do it with one of these. I'll just use the reverse complement, the reverse primer. I'll go back to GenBank, go to the BLAST website again, and put that in there, and we can blast this primer. And we can now blast it against everything in GenBank and see what comes up. So let's do it, for a start, let's do it excluding pangolins. So we can actually look at everything else it might bind to if we hit this little checkbox and select the right taxon. So this is going to look at GenBank apart from pangolin DNA, and it will tell us if there are any perfect matches in there. So this may take a while. I'll pause again. Okay, so the results are back. So that search went in. I would then just look at the taxonomy, and well, there were some hits and other things, but none of the hits were perfect. So although it says 100% identity, it's only over part of the query. So it's a sort of and you can see that if you actually look at the alignment specifically. That's the query primer, and there are mismatches in there. So it's so it's not fully matching it. There's a region here because it's more than 21 base pairs long. That means that is not a perfect match. And if you did the same thing with the reverse primer, you could actually check to see that there were other mismatches in there. So this is a primer set I would go ahead and order. I think that actually looks pretty good to me. I would just buy them because they cost about $60 to buy that primer pair, and start testing them out. I get some pangolin DNA and some other DNA and test empirically to show that it doesn't work. And that's essentially what people do with any of these kind of things, where you like that paper I showed you earlier was where people had empirically tested it. So having shown you that very hacked together way of designing PCR primers, I'll just give you a little bit more information on this. So I actually wrote some software for doing PCR primer design years and years ago, and unfortunately now it was written on a very old version of Windows that now doesn't run anymore. So I mean, I could get this software to run if I wanted to, but it's not really worth it. So I wrote this whole graphically based bit of software. This is the manual for it, which is the only thing I can really access now. It's called Amplicon if you want to have a look. So if you look for Amplicon PCR primer design software and then search through there, you can probably find this manual in there, or I can upload it if anyone's interested. And it does have useful information on the idea of primer design by software. But having said that, I wouldn't bother using it because there's other software available. So this EcoPrimer software is part of the ObbyTools package. So ObbyTools is a package for doing all sorts of things with DNA metabarcoding. It does have this option for it designing PCR primers for you, but you know, it's not necessarily that easy to use, and I actually find doing it the way I do, which I just showed you, is as easy or easier, and you've got more flexibility with it. I guess the only real issue is that you need to have some sort of knowledge about how long ago things diverged and which gene markers are useful for different levels of divergence. Another thing that you could do if you're thinking about doing this is use this. It's not that one. Is use this. Where is it? It's the wrong one. Time tree. There it is. Yeah. So there's this time tree package. This time tree website that is really useful actually for looking at times of divergence. So you can use this to look at how long ago different things separated from each other. So say I was looking at tardigrades again, probably use that name, and how long ago they diverged from jellyfish and other things. Let's see if we can give us a result for that. And once you've done this a few times, of course, you get a bit of an idea of which gene regions would be useful for that. As a general rule, mitochondrial genomes will be useful for a lot of things that like interspecies things, inter-family class phylum things. Then you probably use nuclear 18S or 28S things. So this is giving us a time of divergence for them. Basically, it's a very, very long time ago, so 824 million years ago. So if I was trying to separate those two groups and design PCR primer sets that would distinguish between them, I'd definitely have to use a very slow evolving molecule, and either the 18S or 28S nuclear ribosomal DNA would be appropriate for that. So anyway, that's a pretty kludged together way of how you do PCR primer design. To summarize, first thing you should do is do a Google search and see if someone else has done something. If they have, check to see whether it's useful for you as it is. If not, you might be able to modify it, and you can go through some of those steps that I just showed you to modify their information, and that's worthwhile because DNA sequence databases are often being updated. You know, for a paper like that, tardigrade one was published 11 years ago. If you redid that primer design, it might be better now because there's more information available. If that's not available, think about which sort of molecule type you might want to work with based on the time of divergence for the groups that you're interested in. Then go to GenBank, download some of them, search through the alignments and see what you can find, and then test what you can find to see whether it's good or not. At the end of the day, all of these things need to be tested empirically. So I would really imagine that pangolin primer set would work actually. So if I was going to work on pangolins, I would just order that now, and I'd be 95% sure we'd get some useful results out of that. But you still have to test it. If I was doing that project, I might have a go at designing one or two more. I might look at some other mitochondrial regions and see if I could work with those as well. But in this case, I'm just showing you as a demonstration. So anyway, that's it for this tutorial. I'm working through the bioinformatics classes for next week. I've done 2 of them, 1 more to go. I will distribute the virtual machine for running those classes towards the end of the week to you all. I hope you've seen the lecture on ancient DNA that I produced, and also the lecture on wildlife disease genomics that Dan White produced for us, which was really nice of him to do that. A really interesting lecture, I thought, and some really inspiring stuff, especially given the current situation we're all living under. So anyway, talk to you later. Bye.